Hi, everybody. Welcome to another Chats with Children. I hope you're safe and well. Today, I'm delighted to be joined by Georges Muller, who is the founder and CEO of Seed Biosciences. And George was at SLAS last week. And I have to say, first thing, congratulations, George, because George actually won, or was one of the co-winners of the Product Innovation of the Year Award at SLAS 2022. So we're going to be talking a little bit about that today and talking about lab automation as well. So, George, it's lovely to see you. How are you?
Thank you. I'm very well. How about you?
I'm very well, George. So where are you calling in from today?
I'm back now in Switzerland, in the French-speaking part of Switzerland, as you can hear.
All right, fantastic. Look, I'm very jealous that you were at SLS last week, so I really wanted to go. But congratulations again for the award. But before we start talking about the award and SLAS, would you mind telling viewers a little bit about yourself and also the company?
Yeah, so I'm the CEO of Seed Bioscience and co-founder. We started the business about 2 years ago. That's the fruit of many years of research here in Switzerland, in this Institute of Technologies in Switzerland. Now we are a team of 12 people. We just recruited one this week. So the team is growing. We have a product on the market since about 12 months. Now we have 20 clients using the machine worldwide, in China, South Korea, the US, and in Europe.
Right. So now everyone will be asking the very same question I'm asking now. So what is the product and what does it do?
So we have this new pipetting system, which is called DispenCell. And this system enables scientists to isolate single cells very precisely. So one click, one cell.
Fantastic. Well, before we talk about DispenCell a bit more, as I said, you were at SLS last week. So how was SLS for you as an event? I mean, how was the show?
Well, if you are asking for me, I had a blast, like amazing time there. Like always, it's the third time I'm going to SLAS, and this one was very special. Because like everybody on the planet, we stay for about 2 years stuck at home, and so that was my very first business trip after this pandemic, and I just love it. That's why I'm doing this job.
Brilliant. I'm glad to hear it. Well, let's talk about the award then. So I know SLS do this product innovation award. They've been doing it for a number of years as well. So tell me more about the award and about why DispenCell you think won the award.
Yeah, so as you know, it's not the first award we win at SLAS. I think we won them all right now. So this one was very special because it's the new product award, and the selection is pretty harsh. So I think there was tons of companies applying for the award, and then we ended up with 10 in the selection process. And we had jury members coming to the booth, and we had to convince them of the novelty of the machine, the importance of that machine in the field of liquid handling and lab automation. So it was not easy, but I'm very glad we went through the process, and now I mean it's giving us huge visibility, right?
Fantastic. Well, look, I know it's very difficult because we're doing a chat and we're not actually there with the product demo or anything, but can you describe what the product is and what makes it unique then to other similar products in the marketplace?
Well, I'm sure you've worked with pipettes in the past. At least the community knows pipetting system very well, right? And pipettes are tools that are used to dispense liquids and cells, but it's just a piece of plastic and a pump. Nothing else, right? So in this work, we in the past we've been actually able to make this technology smart, and we have turned it into a pipette which can dispense single cells one at a time. So this is kind of a revolution in the fields, and I think that's one of the reasons why we have so much traction in this space of lab automation, because now we come with a technology, with a pipetting system that can really pipette single cells super precisely and with some traceability as well.
And for the layperson out there, what is the benefit of being able to do single cells? I mean, for a researcher.
So it's a really good question. And now actually in the biology and life science, the capacity to isolate single cells is becoming very much important, more and more important.
Right.
And this is because of the heterogeneity of the cells in general. Like, I don't think about cancer cells, for example. You take a biopsy of a tumor, and in this, basically all tumors will be different from patients to patients. And if you look at the level of the tumor, at the level of the cells, all the cells are different. So there is a need. So there is a need to basically study those cells one at a time and to better understand the heterogeneity, and then define drugs that can target those cells very specifically.
Right, fantastic. Now, as you mentioned, that this isn't the only award that you've won, is it? There's other awards that you've also won.
Yeah, yeah. So I think the first time I went to SLAS was in Washington, D.C. I was still a fresh postdoc out of the lab. We went there on the Innovation Avenue, great place to be, under the spotlights, and then we won the Ignite Award 2018. Then the year after in San Diego, I was selected among the top 10 Innovation Award. And now we have the New Product Award. So I'm very happy to see this continuity and to see the support of the SLAS community.
Fantastic. Well, you're also ideally qualified then for me to ask you this next question. I mean, being at the show this year, I know there's a lot of new products launched. What were the other coolest innovations that you saw at SLAS this week, or last week, I should say?
I was really impressed by the new innovations that are now coming to the market. And what surprised me, but maybe not so much because we had this vision also before, is all the in lab automation, there are many companies that are going in the field of single cell biology because it's booming. I mean, now if you look at the number of publications and the number of workflows that are designed around that, it's very impressive. So many companies are now developing workflows to address those the needs in the field, and they're actually in particular in lab automation, they're now trying to combine different tools to complete the workflow and address those questions of high throughput, traceability, etc., to make it basically GMP compliant and compatible with clinical workflows. So really industrial workflow. And what I like, so it's basically like combining Lego blocks. When you take a block and you combine it with another one, so at the end of the day you have a workflow that is simple to use and makes sense and works from end to end. And so for the final user, it's a super easy because it's plug and play, and then down the road they get the results, they get what they wanted, and it's fully automated. And what I like there is that actually we could see that our technology is a great fit in this workflow. Like it's a important Lego blocks in all this chain, and we have actually lots of tractions from all those different companies that want to integrate our technology into that. So I'm super happy to see that.
Well, that's really interesting you said that, because I was thinking then, one question I've got for you then, as someone who works within the industry and does new technology is, how compatible are all the different instruments now within lab automation to do that sort of Lego block building, if you like, in terms of the sector? I mean, because obviously if you look at lots of technologies when they come out in the marketplace, often everyone's got their own unique standards, and it's very hard to see the compatibility of how they fit together, whether it's the old video recorders, I'm old enough to remember, with Betamax and VHS, and then moving on to that. And everybody wants to have something unique, so they stick to whether it's Apple and Android and so on. So within the lab, how compatible are most instruments now when they're being launched and being able to fit together?
It's a good point, and I think the biggest challenge is to find tools or piece of technologies that are compact, open to be integrated in with other instruments, and affordable as well, because at the end of the day, cost matters for the clients. And if you want to be competitive, you have to come up with added value, obviously, but with the right price. So I cannot speak for the others, but I can speak for ourselves. I think with what we have now, we have a robot which is super compact. It's the size of a A4 paper, so it's very small, and it's basically an open structure. It's like a pipetting robot, so it's open, and so you can basically make sure that if you have a robotic arm, you can take the plates from the robots, from the DispenCell to another machine super easily because it's open. And then we have built some API around that. So yeah, and finally we have the technology which is disposable, so you don't need to have complex machine around it to do the washing steps, etc., etc. It's just single use. So I think that's another advantage, and it's also a very affordable solution.
Fantastic. Well, thank you for answering that. Now, this may seem like an obvious question, but what's been the best outcome for you attending SLAS this year?
Well, if I look at the pile of business cards we have collected, that's the first outcome. I think we have filled the sales pipeline with really interesting leads there, and I'm very happy. I've seen and met so many customers at the booth, and you know I've seen the ratio. There was about 50% of people attending that are actually end users and customers, and 50% exhibitors. And for us, this ratio is just perfect because we can gain leads to sell the machines that we have now, and we can actually do a lot of business development. And we had many companies, you know, I'm not going to mention them, but you can imagine the big guys in the world of lab automation. They came to the booth and they really want to do something with us because, as I said, we have a great piece of technology they can integrate in their own machines.
Great. I mean, I think that's actually true now of a lot of industries, isn't it? A lot of technologies now where since the pandemic, people have been more open to collaborating, whereas before they were quite insular in how they work. But I think everyone is now looking for more collaboration in terms of product development.
Yeah, well, hard for me to say because we started the business 2-3 years ago. So, but I trust you. I trust you, Rizwan, on that.
All right. Well, yeah, trust me on that. Yeah. Right, so looking ahead, I mean, you've won. Obviously, I mean, what's been the feedback? I mean, I know it's only been a week since you won the award, but what's been the feedback to winning the award?
By feedback, you mean like visibility or?
Yeah, the impact. I mean, what's been the feedback to customers, potential customers with you? I mean, has it changed how people have perceived you and the technology in the business?
Yes, I think so. Already now we can see it. Just in terms of lead generation, we've gained lots of traction in the recent days. I'm very happy to see the website contact forms, you know, to work even more than before. So I think that's the first measurement of that success. And then, yeah, I mean, with such an award and recognition from the peers and from the industry, I think this is giving us a lot of credibility as well. We're not just the Swiss guys developing a cool tool. Now we have something very serious in hands, which is available now for customers, and that is a very nice match for the industry. So I think this is nice, and I'm really grateful to the SLAS community and the society for putting that in place.
Brilliant. Now, obviously, you won this award. You mentioned you won the Ignite Award and various other things. So, looking ahead, what can we expect going forward from the company and your technology?
What's
next?
What's the next award, or
what's the next step in the technology and so on? What are you sort of developing going forward?
Yeah, so we're not going to stop there. We have this technology for single cell isolation. So with the so-called impedance spectroscopy, which is not optics, we don't take image or fluorescence. It's just a very elegant and simple method for detecting the cells. It's so-called impedance spectroscopy. So now we give a very simple information. We can say when you click on the pipette or on the robot, how many cells do you have in the well? Is it 0, 1, or 2? And that we can control and trace that very precisely. But in fact, with impedance spectroscopy, we can do way more. and we have research projects in the company where we know we can address questions like cell viability, protein production, etc., in an immediate and non-invasive way. And when I say non-invasive, it's like without labels or anything like that. It's just with using impedance spectroscopy. So this is giving you basically the direction where we want to go.
Right, brilliant. And one final challenge, but obviously as a startup, relatively new startup, in a couple of years, and you've got the new technology. What are the big challenges that you face then in terms of developing the business in the current climate?
Yeah, it's a good point. So obviously we have a good team of engineers. So about half of the team now are engineers. The rest are salespeople and application engineers. So we are really now building the sales structures and sales force in the company. But half of the company of 12 people, it's only 6, so it's not that much. And that's where the bottleneck is now, is to be able to gain visibility, generate leads, fuel the pipeline, and make more sales. And that's where we put all our efforts now to make the clients happy and the investors happy as well.
Right, yeah, well, obviously. Well, look, I wish you lots of success on that then, George. I mean, it's been lovely to see you. I'm very jealous I wasn't there last week, because the more I talk to people who attended last week, the more I'm frustrated I didn't get to go. But I'm glad you went, and I'm glad you won the award. So congratulations again on winning the award.
Yeah, thank you very much, Rizwan. And if I may, I want to make sure we can meet in San Diego next year, and we do the same interview on the booth.
Oh, you know what? I would love to be in San Diego, and I interviewed SLAS earlier this week, and they told me the dates for it. I know it's later in February next year, so I look forward to it. So there you go. Now, one final question, George, for you is that obviously people are listening to this and they're thinking, well, actually, we've talked about this wonderful new technology. One, where can they get more information? And two, where can they see it live? I mean, are you going to be at any events going forward for the rest of this year as well?
Yeah, I mean, if you want to contact us, it's a super simple. We are friendly people, as you can see. So just get in touch with us through LinkedIn, emails, or the website. And the next event for us, so we have a couple events we want to attend in the field. So we have the ESACT meeting. It's a European Society for Animal Cell Technology, great place to go in Lisbon. Then we have the ISSCR, it's the biggest stem cell congress in San Francisco. And then a couple other meetings that we're going to attend.
Right. Are you going to be at SLAS Europe?
I have to think about it.
Ah, right. Okay. Well, hopefully you're there. If you are, then I'll hopefully maybe see you there as well. We never know. All right. Well, look, George, thank you very much. That's been great to learn more about the technology and your success last week at SLAS, and also your views on SLAS as well. So thank you very much for sharing that. So there you go, viewers. If you'd like to know more about George's technology, then please get in touch with George. You can get in touch with him via LinkedIn. You can go to the website. You can meet them at the shows. There's loads of ways to contact. And if you've got any questions for George, you can leave them in the comments below the video as well. So feel free to leave any thoughts or comments that you have, or even congratulations. I'm sure we'll be happy to see them as well. So put them in the comments below. So George, all the stuff he says, I wish you lots of success with the product. Have a fantastic 2022 and follow up on all those leads. I mean, I'm terrible. I go to shows, meet lots of people, and I always forget to send them information. So I'm the worst at following up leads. So make sure you don't do that, and have lots of success for the rest of the year. It's lovely to meet you again. And there you go, viewers. If you'd like to know more, as I said, check out the links. And until next time, as always, stay well and stay safe. Bye-bye.
Bye.